Hello everyone, welcome to Study Biology with Jisha. Today we are going to have a very interesting video for class 12 as well as for advanced class 10. So stay tuned and watch on. So today I will be helping you to prepare a working model of Lac OPERON. Class 12, you have a great deal. So let's start making the working model of class 12 Lac OPERON, and it will be interesting for you, it will be time engaging, and it will be very important for your understanding of Lac OPERON. Hope you like the video. If you like it, please share and subscribe my channel.
Hello students, so today
Study
we are
Biology
going to
with
do an
Jisha.
activity on Lac OPERON. So as we all know that Lac OPERON is carried out in case of E. coli, that is a prokaryote, and it will by using this Lac OPERON we can understand how the structural genes, that is ZYA, carries out its function. So for understanding the operation or the functioning of a Lac OPERON, I have designed a very interesting activity which everyone can do it at their homes, and this activity requires a board which can be an exam board, a A4 size paper, sketch pen which is black in color, and many color pens are here also available, some crayons, ruler, a pencil and an eraser. So with these things and other things are also required. With the course of time I will be telling what stepwise what are the requirements. So in the first step, what I have done is I have taken a exam board. On that I have placed this paper and I have drawn the operon as given in the NCERT book. So as you can see, this by using this sketch pen, I have drawn this with a ruler and I have shown the promoter, the regulatory gene, the second promoter, operator, and the structural genes Z, Y and A. Now what is the next procedure I will be following? So now what I have planned is I will be coloring all this in different colors. So as you can see, 1, 2, 3, 4, 5, 6, 7 parts are there. This is an operon, this is a transcriptional unit. So every part I will be coloring differently. So let's start the process. So let's start with the first one with green. So you must be wondering that a biology teacher means it should be the person should be very artistic. No, the person should be imaginative, so that the processes which they are studying, that can be easily done at home, and which can create interest among yourself. So and you can also understand concepts. So let's see, I have already colored the first promoter in this light green color. Then I will be coloring with I just love light colors, so I'm taking this one. So this regulatory gene I am coloring as pink. Okay, so this regulatory gene is now pink in color. So similarly, you are going to color all these parts, all these segments and genes in different colors. So this promoter I am coloring brown. This is the operator region. so now it's done. So this is now predicting the Lac operon. So this Lac operon has 7 different segments, and each of them are, of course, this is a transcription unit, so each of them is a single gene. So this is the first promoter gene, followed by the regulatory gene, second promoter, operator, and the 3 structural genes ZYA. This is step 2. In the step 2, what you're going to do is you are going to cut open this operon with the help of the scissor. So I have brought a scissor, so I am just now cutting it down and actually trimming it so that we can use this operon in our next procedure which are going to come. Okay, so this is my operon. Not a problem if I keep extra papers surrounding it, because it may be helpful for me. So just trim down. Okay, so this is our operon ready. Now what I am going to do is the next step. Alright, so now what I have done is I have laminated this operon. How did I laminate it? You must be wondering. So I have taken this tape and I have put this tape on both the sides and I have laminated it. So after this, now why have I laminated it? So that my procedure will be very easy. That is why I have laminated, and it will not cause any bending of this sheet, and it makes the sheet also very tough. So after laminating this sheet which is having my operon, I have to remove this extra parts. So now I am removing this extra parts which is now not required. This extra parts was given only to facilitate my lamination and cutting process. So I have cut down my extra parts. So this is now the whole operon which is ready with me. Now what will I do is my next procedure. So now let's start the next step. In the next step, I have taken this beautiful, interesting kids thing, which is the fundo, and this has many colors. Out of these all colors, I have chosen 2 colors. One is purple and I have shaped it like this, and the other one is green and I have shaped it like a ball. This green color, which is having some portions of pink also, because it is a mixed color, so this pink color will act as a repressor protein. So the repressor protein is produced by, or is rather translated from the repressor mRNA, and the repressor mRNA is transcribed from the regulatory gene. So this regulatory gene has transcribed repressor mRNA, and that repressor mRNA has given rise to this repressor protein. And this purple color one, which, or the violet color one, whatever you call it, will act as the RNA polymerase. The RNA polymerase, which is going to facilitate the transcription of these structural genes ZYA. Okay, as we all know, now this operon is not functioning. Now we will start the functioning of it. So for the functioning to be carried out, we all know that first always the operon is in switched off condition. So what is the meaning of switched off condition? In the switched off condition, this repressor protein, what does it do is it comes and attaches itself to the operator. So as you can see, I am attaching the repressor protein to the operator. Okay, so the repressor protein has attached itself to the operator. Now I am attaching the RNA polymerase with the promoter site. So this is the RNA polymerase which is getting attached to the promoter site. Okay, now the problem what you can see is this RNA polymerase is not able to shift. Is it shifting? No. Why? Because it is being blocked by this repressor protein. This repressor protein is attached to the operator site and is not able to carry out or facilitate the mRNA to carry out the transcription of the structural genes Z, Y and A, and hence this condition is known as the switched off condition of the operon. Alright, so this is known as the switched off condition of the operon. Now for making it switched on, we require a inducer. This inducer is carried out by one interesting thing which is known as the lactose. So this paper clip is taken as the lactose. Now what will the lactose do? When lactose is given to the system, this lactose is attached to the repressor protein. So when the repressor protein is produced by this regulatory gene after transcription, then by the translation, it will get attached to this paper clip. What is the paper clip here? The paper clip is the lactose. So hence when the paper clip is attached to it, so now the problem is, you can see the bits are falling down. Okay, so this is inactivation of the repressor protein. So now because of the binding of the lactose to the repressor protein, there is inactivation of the repressor protein, and hence, as you can see here, the operator site is free. So what will happen now? Because the operator site is free, this promoter can easily start the process of transcription and is now easily penetrating through the structural gene. As you can see here, see, it's penetrating. One more time I can show you. I'm just removing it and I'm showing you again how it is penetrating. So see, from here, from the promoter, this is the purple one is the RNA polymerase. The RNA polymerase is carrying out the process of transcription of the structural gene by flowing towards it. Okay, so this is how the RNA polymerase carries out the transcription. Once the RNA polymerase carries out the transcription, then the LAC mRNA is produced. This LAC mRNA will carry out translation and will produce three important proteins. One is Z gene, which will code for beta galactosidase, Y gene, which will code for permease, and A gene, which will code for trans-acetylase. So this is the switched on condition of the Lac OPERON. Hope this activity would have been very interesting for you. You can also try this at home and explain any student around you. And this is very interesting for you all, so that you can, adapt this and get engaged in such activities. I hope you can find out such simple working models for RNA, your DNA replication, for your translation of RNA, for transcription of DNA to RNA, and can easily enjoy each and every bit of biology. Thank you and have a great day ahead.